Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go. Let's make great science together.
Okay, let's dive right in. For years, maybe decades, everyone's been chasing these protein targets, or, well, parts of proteins, that just refuse to sit still.
The intrinsically disordered regions, IDRs, the floppy bits.
Exactly. And they were basically written off, right? The ultimate undruggable targets. If it's like shapeless spaghetti, how do you even start to design a drug for it?
You don't, or at least that was the thinking. No stable pocket, no keyhole to aim for.
Right. But here's the wild part. The real breakthrough. Scientists have figured out how to design completely new, stable proteins that don't just tolerate this disorder, they actually, use it.
They exploit the flexibility, and they're getting super specific, super tight binding. We're talking picomolar levels.
They essentially turned undruggable into designable. That's the core story here.
And it's a huge deal because IDRs are everywhere in biology. They run signaling, transcription, phase separation, really crucial stuff. But that constant shifting shape made them impossible targets. Until now, maybe.
So that's our mission for this deep dive. Cut through the jargon, unpack this new protein design playbook they call Logos, and really understand how they managed to engineer specificity against this kind of biological messiness.
Yeah, and show why it's not just a cool trick. It has immediate applications, like right now.
Okay, so let's set the scene a bit more. Traditionally, designing a drug or a binder, you're looking for a nice, stable pocket, right? A defined shape on the target protein.
Exactly, like designing a key for a very specific rigid lock. And, previous de novo protein design methods got really good at that, provided you had a crystal structure.
But IDRs,
Yeah.
no lock. It's more like trying to grab onto smoke. There's no single structure to target.
Right, so the whole philosophy had to change. Instead of designing for a rigid target,
They designed binders that force the flexible IDR into a specific shape, like an induced fit.
Exactly. Induced fit, but maybe on steroids. They designed the binder to recognize certain features along the IDR sequence and basically coax it or, force it into a stable bound state that might not even exist otherwise.
Okay, so how did they actually do that? Especially for complex, real-world IDRs, not just simple repeats.
That's where the cleverness comes in. They blended the old and the new. They used classical physics-based methods, think Rosetta, which are great for designing stable structures.
Okay, stability.
Makes sense. But they combine that with the power of modern deep learning, specifically a tool called RFdiffusion, which is fantastic at generating diverse and novel protein shapes.
So physics for the foundation, AI for the creativity and complexity.
You got it. And this combined pipeline, which they named Logos, is what let them build this sort of universal playbook for disordered targets.
Let's walk through that Logos pipeline. Step one.
Step one was scaffold generation. They started with the basics. Using Rosetta, they designed these simple, repeating protein scaffolds. Think of them as like basic building blocks, very stable.
And these scaffolds were designed to interact with peptides in what kind of shape?
And an extended conformation, because that's how IDRs often interact naturally stretched out with their side chains exposed.
Okay, so the repeating scaffold, maybe made of simple units like LK or YD repeats, could form hydrogen bonds with the backbone of this extended peptide.
Precisely. It gives you a stable base interaction, kind of independent of the specific side chains at first.
But like you said, real IDRs aren't simple repeats. So how did they get specificity for complex sequences?
Right, that's the limitation of just repeats. So steps 2 and 3 involved pocket specialization and assembly. This is where it gets really cool and where deep learning comes in.
Okay.
They essentially took the parts of those simple scaffolds that formed potential binding pockets, spots that could recognize, say, a specific amino acid side chain.
Like modular pieces?
Exactly like modular pieces. And then using RFdiffusion, the AI tool, they stitched these specialized pockets together into totally new chimeric backbones.
Whoa. So mixing and matching the best bits to create something completely new and tailored.
You nailed it. It's like taking specialized tools from different toolkits and combining them to build a unique machine for a very specific, non-repetitive job. This generated a huge library, 1,000 unique template structures.
1,000 templates. And these weren't limited by simple repeats anymore. They could, in theory, recognize any arbitrary sequence.
That's the key idea. They built a diverse library capable of handling the complexity of real biological IDRs.
Okay, library built, what's the final step? How do you find the right binder for your specific IDR target?
That's step 4, threading and refinement. It's actually very efficient. You take your target IDR sequence, let's say 15 amino acids from some protein you care about, and you computationally thread it through the entire library of 1,000 templates, looking for the best structural or geometric match. Which template best fits this specific sequence?
Okay, so find the best starting point from the library.
Right. And once you find a good match, you use computational tools, again, machine learning included, to refine the binder's sequence, optimizing all the contacts between the binder and the target IDR.
To really crank up the binding tightness.
Exactly. To push the affinity into that really strong picomolar to nanomolar range. It's a very focused process, way faster than experimentally screening millions of random variants.
Okay, the computational strategy sounds powerful. Very elegant, really. But the proof is in the pudding, right? Did it actually work in the lab?
Oh, it worked. And the success rate is what really makes you sit up and take notice. They didn't just test it on 1 or 2 easy targets.
No. What was the scope?
It was pretty ambitious. They threw 43 different targets at the Logos pipeline. That included 18 synthetic peptides, just to cover different chemistries, and 21 diverse biologically relevant IDRs.
21 real IDRs. But what?
Things like bits of transcription factors involved in cancer, neuropeptides, a real mix of challenging targets.
Okay, 43 targets total. And the success rate.
Get this. They successfully designed and validated binders for 39 out of those 43 targets.
Wait, 39 out of 43, that's like 90% success for de novo design against IDRs.
Yeah. It's kind of jaw-dropping.
Yeah.
If you look back at earlier protein design work, especially for hard targets, getting even close to 50% was considered a big win. Hitting 90% on the first try computationally is,
Well,
it's transformative.
and how much effort did it take per target? Were they testing thousands of designs?
That's the other amazing part. On average, they only needed to experimentally test about 22 designs per target to find a good binder.
22, that's it?
Yep. Compare that to screening huge libraries. This is much more like rational, targeted design. Almost one-shot success in many cases. Huge speed up.
Okay, so high success rate, high efficiency. What about the binding itself? Strong, specific.
Both. The affinities were often extremely high down into the 100 picomolar range, sometimes up to maybe 100 nanomolar, but consistently very tight binding.
Which is great, but pointless if it sticks to everything, right?
Yeah.
Specificity is key.
Absolutely crucial. And they check that rigorously with what are called all-by-all binding matrices, basically testing every binder against every target. High specificity. The binders only bound strongly to the specific IDR sequence they were designed for. Even when they tested against very similar sequences, the binders could tell them apart. That's orthogonality.
That's impressive. Did they push the boundaries in other ways? What about really difficult types of IDRs?
They did. They specifically went after highly polar IDR sequences loaded with charged or polar residues like glutamic acid, lysine, serine. These are notoriously hard for design because the interactions are so complex and water-mediated.
How polar are we talking?
Some targets were over 75% polar residues. They even designed a binder for a fragment of the EWS-FLI oncoprotein, which drives Ewing sarcoma. That target is 84% polar.
Wow. And they got binders.
They did. It showed the method is general enough to handle these really challenging chemical environments by designing scaffolds that could make the right hydrogen bonds to manage those polar interactions. Big technical achievement.
So we've got the numbers, high success, high affinity, high specificity, even for tough targets. But what's actually happening structurally? Did they confirm that induced fit idea?
Yes, and the dynorphin A example is the perfect illustration here. Dynorphin A is a neuropeptide. It binds to the kappa opioid receptor, or KOR. It's naturally flexible.
Okay.
They used X-ray crystallography and NMR powerful techniques to see structures, and confirmed that their design binder forces dynorphin A into a single, stable, well-ordered shape when bound.
So the floppy peptide becomes rigid upon binding. The induced fit is real.
Exactly, but here's the really fascinating detail. The structure of dynorphin A when it's bound to their designed binder is different from the structure it adopts when bound to its natural partner, the KOR.
Hold on. So the design protein forces the IDR into a shape that nature doesn't typically use for its function.
Precisely. It shows that computational design isn't just limited to copying nature. It can actually sculpt these flexible molecules into completely new, non-native conformations.
That opens up, wow, a lot of possibilities for manipulating biology in new ways.
It really does. And they also showed incredible control over specificity with dynorphin. There's a short sequence at the beginning of dynorphin A, YGGF, that's shared with other related peptides.
Like dynorphin B.
Exactly. Dynorphin B is very similar, like 62% sequence identity, but they specifically designed their binder to ignore that shared YGGF motif.
Clever. So the binder only recognizes the unique parts of dynorphin A.
Right, and the result was it bound dynorphin A tightly but completely ignored dynorphin B, despite the similarity. That level of fine-tuned selectivity is exactly what you need for clean biological tools or therapeutics.
Okay, so the mechanism is confirmed, the specificity is there. How useful are these binders in, actual biological systems? Do they show applications?
They did, across several really important areas, showing these aren't just test tube curiosities. First, proteomics and diagnostics.
How so?
They designed a binder for a specific short peptide, a tryptic fragment from a protein called cystinosin. This peptide is normally found at very low levels in blood.
Hard to detect, typically.
Very hard. But they used their design binder to successfully fish out and enrich this low abundance peptide directly from blood samples.
That's huge for diagnostics. Imagine being able to rapidly design cheap custom affinity reagents for any biomarker peptide you want to measure.
Exactly. It could really change how we approach biomarker discovery and low-cost diagnostics.
Okay, diagnostics. What else?
Cell targeting.
Yeah.
They designed a binder against mesothelin, MSLN, which is a receptor found on the surface of certain cancer cells.
And did it find the right cells?
Perfectly. They showed the binder stuck specifically to the surface of cancer cells that expressed MSLN, but it completely ignored control cells that didn't have the receptor.
Wow. So highly specific targeting, even in the complex environment of cell surfaces. You can imagine using these for drug delivery or maybe CAR-T therapies?
Definitely points in that direction. Then they circled back to the therapeutic potential with dynorphin A.
Did the binder actually block its function?
Yes. They used a cell-based assay that measures KO signaling. The designed binder effectively inhibited that signaling pathway by preventing dynorphin A from activating its receptor. They got an IC50, a measure of potency of 50 nanomolar.
So it's not just binding, it's functionally interfering. That's a preclinical proof of concept.
It is. And maybe most importantly, they showed these binders work and maintain their specificity inside living cells.
How do they test that?
Using something called a mitochondria colocalization assay, they basically tagged the binders and their targets to see if they would find each other inside the cell.
And does they?
Only when the correct binder was paired with its intended target. Perfect orthogonality, even within the crowded, messy environment inside a cell. That's a crucial demonstration of robustness.
Okay, so summing this up, the design works, the success rate is phenomenal, the specificity is high, and the applications are immediate and diverse. What's the big picture innovation here?
I think it's the scale and the generality. This isn't just one clever trick for one specific IDR. The Logos method seems to be a robust, broadly applicable platform. It really changes the game for targeting disorder.
And the key advantage comes from kind of embracing the floppiness.
It seems counterintuitive, right? But yeah, because the target IDR is flexible, it samples many different shapes or conformations. The design pipeline can explore this conformational landscape computationally and identify conformations that are compatible with very tight binding, even if they're rare or transient normally. In a way, the flexibility gives the designer more potential solutions to choose from compared to a rigid target that might only offer one or two binding sites, if any.
So disorder becomes an advantage for the designer. That's a cool twist.
It really is, and the implications are pretty significant.
Like what? Which doors does this open?
Well, think about all those previously undruggable disordered proteins involved in disease. Major cancer drivers like the EWSFLI fusion protein we mentioned, or EML4, ALK in lung cancer, maybe even specific RAS protein isoforms that have proven so hard to target.
Suddenly they look more accessible.
They look a lot more accessible. And beyond direct therapeutics, this platform is basically a factory for generating new biological tools.
Custom tools. Like what?
Highly specific biosensors to detect tiny amounts of specific peptides, Reagents to modulate or even visualize liquid phase separation, those protein condensates involved in neurodegeneration and other processes, plus, like we saw, tools to supercharge proteomics by enriching specific targets.
And where could the method itself go next? Can they refine it further?
Oh, definitely. One obvious direction is designing binders that are sensitive to post-translational modifications, or PTMs.
Like phosphorylation. So you could design a binder that only recognizes the IDR when it's phosphorylated, for example.
Yeah. Exactly. That would let you specifically target the active form of a signaling protein or track changes in PTM states within a cell. That's incredibly powerful.
That would be amazing for research, and maybe even diagnostics or therapy.
Another, maybe more ambitious idea is incorporating catalytic activity. If you can design a protein to hold a flexible target in a specific shape.
Could you build in an enzyme active site right next to it?
Potentially.
Yeah.
Maybe design a custom protease that only cleaves the target sequence when it's held by the binder. A general way to achieve targeted protein degradation, perhaps.
Wow. Okay. So many possibilities unfolding. We've really covered a lot of ground here. We took a deep dive into how these researchers basically rewrote a major rule in protein design, making these messy, flexible IDRs finally designable.
Yeah, showing that what looked like an impossible challenge, the disorder itself, could actually be turned into an advantage with the right computational approach.
It really hammers home that shift in perspective, doesn't it? The flexibility and apparent randomness we see everywhere in biology isn't always a problem to be engineered away.
Maybe it's an inherent feature, a strength almost, just waiting for us to figure out the right geometric language, the right computational tools to interact with it productively.
So maybe the final thought for you listening is this. If we can now engineer proteins to grab onto nature's most flexible molecules and force them into specific, even non-native shapes, what other biological problems that seemed impossible because of their complexity or flexibility might suddenly be within reach?
Right. What happens when you can design not just for structure, but for controlled interaction with dynamic disorder itself? It feels like a whole new chapter might be opening up, something to think about.